Good afternoon, sir.
Good afternoon.
Good afternoon, sir.
Good afternoon. Meanwhile, all joins, so we can have a recap of what we have done yet. So we were in our module number 1, and we were talking about different enzymes. And in that, first we discussed what is the history that by which we reach to whatever the status we are. And then we talk about the tools and what are the different tools which we are going to use in the recombinant DNA technology. And as mentioned that there are 4 different classes, we have classified them depending upon their usage. And out of them, we were talking about the first one, that is the nuclease, and the nuclease one is which is going to break the phosphodiester bond, or simply enzyme which is breaking the nucleic acid. The enzyme whose substrate is nucleic acid, and it is converting into the nucleotide. So these are the nucleases. And then we classify on the basis of how they are going to act, which phosphodiester bond they are going to break. If they are working at the ends, we are calling them exonuclease, and within we call them the endonuclease. And here are some example of them: Bal31, exonuclease 3 of exonuclease. And within the exonuclease, we also talked about where they are producing cut, at which end they are producing cut. They are producing cut only at both the ends, or they are producing cut only at the one end, that is the 5' or the 3'. Then we are talking about the endonuclease, and the endonuclease were the enzymes which are cutting within. Means at the after 2 nucleotides, simply in simple way we may say that are the endonucleases. And we gave the examples whether they are producing single-stranded, creating single-stranded one or the double-stranded one. So example which is producing cut in the single-stranded one is SI nuclease, and double-stranded one is the DNase. And more type of the endonucleases are there which are producing cut at a specific site or a limited of sites. To them we call the restriction endonuclease. And this is a further the second that we already talked about. And then one which is the main, and on which we stopped in our previous lecture. And this is about the enzyme that called the restriction endonuclease. And these are the most important enzyme in your recombinant DNA technology, or simply say the molecular biology, because this is the real genetic engineer. This is the one which is really engineering your DNA. Without this enzyme, we can't stand where we are. And any of the experiment which we are going to perform in your laboratory, most of the experiments are going to be based on this property of the enzyme that we call the restriction endonuclease. Okay, so this is the one what we have done yet, and now I think most of you are also here, so we can proceed. Okay, so here is a discovery how these enzymes being discovered, because we are just naming them. These are restriction, these are the endonucleases, these are the exonucleases, because till this point it was known that enzymes are there and they are the nucleases, but they are cutting a specific location. That how it comes into the picture. Okay, so these types of enzymes, or the restriction enzymes, that the name restriction is there. The name restriction comes from that they are restricting. What they are restricting? They are restricting the growth, they are restricting the entry, they are restricting the propagation of bacteriophages, or simply you can say the viruses. These restriction endonucleases were the immune system of bacteria. Means these are the enzymes which protect bacteria from the infection of phage, or simply bacteriophage. So these are bacteriophage, and the bacteria are behind the discovery of these restriction endonucleases. So in initial observation that was eventually led, the restriction endonuclease was made in 1950, when it was shown that some strain of bacteria are immune to bacteriophage infection. So this is the main key behind the discovery of your restriction endonuclease. That bacteriophage, you all know that these are the phages which are in, these are the viruses which are infecting bacteria. But it was come to know that some strains, here we are not referring the species, here we are just referring the strains. Like you all know that the E. coli is there, and different E. coli strains are there. Might be in your microbiology class, when Dr. Devendra Pai might teaching you, they mentioned you that E. coli DH5 alpha. So that is the name of the E. coli strain. Similarly, different strains are there which are known, those are of E. coli. So what it was observed, that it was observed that out of, like, if you are having 100 different strains, you found that 5 strains are there which are resistant to the viral infection or the bacteriophage infection. Now, simple my question, they are not different species. When the species are there, then the genetic difference is so vast, then the recognition about the virus may be different. So the host virus infection may going to be changed. But here they are the same host is there, only some strain is different. Strain is very minute differences are there. Specifically, both are the both same species. Okay, so that's why those bacteriophages which are uh, immune to the, sorry, those bacteria which are immune to the bacteriophages, that phenomenon is named as host controlled restriction. What it means is host is controlling the restricted entry of the bacteriophages. So this is the one, and from this the system came that we call the RM system, which came into the picture. What this R and M means? R and M is a property of your enzyme that you are talking, the restriction endonuclease, or when you are referring the immune system of your bacteriophage. So, sorry, immune system of your bacteria. So bacteria immune system, that immune system, the system is there in which two types of the enzymes are there, or not just the two types of enzyme, two types of the enzymatic activities are there. One is called R, that R stands for the restriction, and second is the M stands for the modification. So these two properties are there, which make the immune system, immune response, or simply what we are saying, immune system of your bacteriophage. So what this R is there? R is the restriction. Restriction means a site-specific restriction. Restriction means a site-specific endonuclease activity. Endonuclease means you are cutting within the your DNA, but it is restricted. The sites are restricted. Uh, Only at the restricted site it is going to produce the cut. Okay, so a site-specific restriction endonuclease that is responsible for digesting exogenous DNA. So here the term is the exogenous. That's why we are calling the term immune. So it is digesting only DNA which is coming from outside. Okay, so that DNA which is coming from outside, your system recognize it, like as in our case, our immune system recognize a foreign body and start producing immune system, immune response, like cellular immune response or human immune response that you might study in your course, immunology courses. So similarly, bacteria also produce an enzyme that is called restriction enzymes, which chop off the DNA, which is exogenous in nature. So now the now simple question came: how your bacteria is going to distinguish that this DNA is exogenous? That you all know that virus is exogenous in nature, but how bacteria is going to recognize that this DNA is exogenous? Now, in case of immune response, we are saying that the antigen is a different protein are there, different protein structures are there. Those proteins are present in the pathogen which are not present in the human. That's why the known self and known self concept is there. But in case of DNA, it is just sequence. It is just DNA sequence. All the nucleotide GATC, all GATC are same, not just similar, they are same. So now how they are distinguishing? So this distinguishing property is coming from the next one that we're talking about, the M. M stands for the modification. Modification means what type of modification we're talking about. So here the modification enzymes that we will refer later in study, that is called the methylase. So these methylase, the term methyl is there. So it is substrate of this enzyme is going to be methyl group, or we also call it as a methyl transferase. Okay. So these enzymes are going to transfer methyl group to the your self nucleotides. Means the DNA that your bacteria is going to have, which is its self DNA, it is going to be methylated. Okay, so means the DNA which is going to be methylated will be recognized by restriction as a self, and DNA which is not methylated will be recognized by your restriction as a non-self, and it is going to produce cut on the DNA which is non-methylated. So this is a simple way. It is a simple your this is a simple phenomena in the how the this restriction in discovered. So DNA with identical sequence specificity, which is responsible for modifying and producing endogenous DNA from similar digestion by the restriction. Now, how this came to know that such type of the modification is there? So this came into the knowledge by simple experiment. When you infect one virus, initially when you're just infecting the virus, by bacteria with the virus, that bacteria is going to lyse it. Okay, so but after some time, the initial titer, viral titer is going to be low, but some virus may evade also this type of things, and they survive and they establish the infection. So when they establish the infection, the property that the modification property is there. So during this time, your bacteria start behaving that virus also self. Then how it behaving like itself? This modification property, it is also modifying the viral DNA. So if viral DNA is also modified and it also contain the methyl group, then it also will be recognized as a cell. So what is going to have? So E. coli C strain, C is a strain name plus lambda phase. We are going to find that lambda low phase growth. So lambda phase growth is going to be low if you inject lambda phase into the E. coli strain C. But when you keep it in there, what is going to occur? Next endonuclease again methylating the viral DNA. Then what is going to happen? Now this low growth lambda phase, if you isolate from there and you infect the same E. coli C strain, fresh E. coli C strain with such type of the lambda phase, what you are going to find a high phase growth. So high phase growth is why? Because now the DNA of lambda phase is also methylated. It means it is also not protected. But now this methylated DNA, if you inject to the new strain, another strain that is E. coli K strain, so this K strain is also going to, it won't recognize it as a self one, but it will recognize a non-self because the modifications are not at the same site. The E. coli C might having the different restriction enzyme, E. coli K might having the different restriction enzyme. So depending upon the basis, what type of restriction enzyme is there, but the site that is recognized by the E. coli restriction enzyme C may not be recognized by the E. coli restriction enzyme K. So this is the brief thing how this restriction enzyme is being discovered. And now, as we mentioned, that the property of endonuclease, first important property in the endonuclease is the recognition site. So, what do you mean by recognition site? How many recognition sites are going to be there? Recognition site, simple way we can define recognition site. Recognition site is a specific location or a specific nucleotide sequence. So when you're talking about in DNA, the location means nucleotide sequence, where that nucleotide sequence is present. So recognition site for your restriction enzyme is a DNA sequence that your double-stranded DNA, where your restriction endonuclease are going to break or are going to digest or going to break the phosphodiester bond. Okay, so simple way you can say that A, T, G, C, 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 whatever the sequence you name it. So it is the most standard. Now, at what location is going to break? That location, what DNA sequence is there at that location, that is called the recognition site of your resting endonuclease. This is the first basic property of your resting endonuclease, that recognition site. As we are talking about in our immune system, in case of our immune system, we are referring that one antibody, one epitope is there. So no other antibody is going to recognize another epitope. So similar type of specificity is there in case of your restriction enzymes are also there. One restriction enzyme recognize its specific sequence, specific DNA sequence. Your restriction endonuclease won't recognize any other sequence. So if that sequence is not present in your DNA, then your restriction endonuclease is not going to take it. Okay. So now, what type of this sequence is going to be? So now, simple point. Now we ask. We are talking about that this recognition site is going to be a DNA sequence. So what type of DNA sequence? What are the properties of that DNA sequence is there? So what type of DNA sequence? We all know that DNA simple means ATGC, and this ATGC are arranged in different fashion, like AAA, one like your genetic code. You know that if you are talking in terms of triplet code, 64 type of the triplet sequences are there. But here we are not just talking about triplet. It may be of 4 nucleotide, it may be of 8 nucleotides, 5, 6, 7, 8. So usually recognition sites, it is of 4 to 8 base pair long. Okay, so now you can calculate. So what are the probability of the finding this sequence? As you mentioned that in case of the genetic code, when you were referring, you found that 4 raised to power 3 is going to be your basis how many codes are going to be there. So that's why 4 is going to be there. 3 sequence is going to be there in your code. So 4 is to power 3, you found that the 64 base pairs, 64 codes are there. So similarly here also we can find the probability that how many base pairs are possible if you combine 4 nucleotides. So here are going to be 4 raised to power 4. Those 4 are the ATGC and the 4 times combination you are going to be there. So if your DNA sequence you are talking about, then there are 256 possibilities are there which are having combination of 4 nitrogenous bases. So it simply means if you are having 256 base pair long DNA sequence, then the probability of finding a said sequence is going to be 1. Here we are talking term probability. Maybe 2 may be there, but the probability of 1 is there, or maybe 0. So, but probability 260 base pair long DNA sequence you are going to have, there will be one sequence which match your sequence of interest. Okay. So now another possibility, if you say that for 5 base pair sequence it may be recognized, then it is going to be 4 raised to power 5. If you say 6, then it is 4 raised to power 6. So another example of 4 raised to power 6, 4 raised to power 6 is equal to 4096 base pair. So this means if your recognition site is of 6 base pair long, then one recognition, the probability of finding one recognition sequence in 4096 base pair is going to be 1. So if you are having this much long DNA sequence, then the probability of finding of any single combination is 1. Then if you are having 8, so the similar formula you have to put there, 4 is to power 8, 4 is to power 8 will be the 6, 5, 5, 6, 3 base pair. So here the probability further decreasing. So depending on your requirement, if you found if you want to produce more cut, then you can use an enzyme which is 4 restriction, sorry 4 nucleotide base sequence. If you want, then W256 you may get one break. If you don't want so frequently, want mid frequency, then you can choose 6 base pair long gene sequence. Then the probability is going to be 4000. And if you found that you just want to produce few cuts in your whole chromosomes, then you need to go to the octa 4 base. So on the basis of how many, what is the recognition sequence, how much is the length of the recognition sequence, we call them cut. Like these are the cutters. These are cutting. These are breaking your DNA sequence. We call them cutters also. So if
Excuse
the
me, sir.
recombinant site is 4 base pair long, yes.
Sir, there is a lot of background noise that I can hear.
So background noise. Any mic is there on? No. So might be everyone facing this background noise. Janvi,
Yes, sir.
Yes, sir.
Yes.
background noise is also.
Yes, sir. Sir,
Is it now down or still the same background noise there?
so your voice is audible. After your voice, some noise is coming.
So at my point, there is no noise. I can't say anything how it is going to you. Is it better now?
Yes, sir. Now it's clear.
Sir, can you please explain the second and third point once again?
Okay, so the main thing here we are talking about when we are talking about these enzymes, we are talking about restriction endonuclease. They are producing they are they are producing cut at a specific location. So what that specific location is? So how long this sequence of recognition is going to be there? What specific location is there? So we mentioned that one property of this site is there. It may be 4 to 8 bases long. If your restriction endonuclease found such sequence which is 4 to 8 base pair long, it is going to produce a cut.
Sir, again
Okay.
there is
So
background noise.
then we calculate the my screen there is no background, no one is speaking. Even Dr. Pai was sitting beside me. He is also now white. He's also not speaking. Then I can't explain how this background is coming. So, and might be some network issue maybe there. Is it better now?
I think it's better, sir.
Okay. Okay. So is this recognition site is clear?
No, sir.
Yes, sir, it's clear.
Hmm?
No, sir.
Okay. So on the basis of what sequence they are recognizing, you call their name. As the resting enzymes I mentioned even our yesterday's class, they are called cutters because they are producing cut within the DNA. They are fragmenting them. So if you produce a cut, they are producing two fragments. So then you also call them cutters. So based on their recognition sequence, whether its recognition sequence is going to be 4 base pair, 6 base pair, 7 base pair, 8 base pair, we call them the tetra, penta, hexa, hepta, octa cutters. Tetra cutter means it is going to recognize a 4 nucleotide long sequence. Okay. So now another property, we come to the next property of your recognition site is the palindrome in nature. Okay. So it is not true for all the recognition site, but for most of the recognition site, they are palindromic in nature. So what does palindrome mean? Palindrome means read same, or means whatever you are reading left to right or right to left, both are going to be same. Like if you are saying A, if you read from left to right, then also it is going to be A. If you read from the right to left, it is going to be A. Another thing, G A T T C C. Now G A T T C. Now if you complementary of it is going to be again from the another direction G A T T C. So both the sides you are reading same. So that's why called they're the palindrome. So there are the two type of palindrome in it. One you call the mirror palindrome. It's the same sequence that you are referring. Example, if you see here, this sequence is written G T A T G. Okay. So what we can do, just I'm taking pen. Okay, so if I draw a line here, GTA this direction, and in this direction it is again GTA. Okay, so this is a one type of palindrome, means you are reading both same in the both the direction. If you are reading in this direction or reading in the backward direction. So this is what type of the palindrome that we call the mirror-like palindrome, because both the sides are the mirror image of each other. Second is called the inverted repeat palindrome. So here the term inverted. So example, just a moment. So example here you can see this is a G T A T A C. So this is a 5' end. This is the 3' end on the one strand. So they are not mirror palindrome. So because both are like if you read within on the same strand, they are reading different. But on the another one, inverted means the inverted means on another strand. So here also this is 5' and the 3'. Read it GTATAC, GTATAC, GTATAC. So on the reverse complementary strands, if you are sequence, you are reading in the same sequence, that you call the reverse palindrome or inverted repeat palindrome. So reverse palindrome and the inverted repeat palindrome both are the same thing. So there are two types of the palindrome. One is the mirror palindrome and second is the reverse palindrome. Two types of sequences, palindrome sequences may be there in the representation side. So two property we discussed here. First property is that they are going to be 4 to 8 base pair long. Second, they are palindromic in nature. They are reading same in the both the direction, from the 5' to 3', 3' to 5', or on a reverse way, mirror way, or the reverse way. The main question here is you have to read same in the both the direction. Okay, so among these two types of the palindromes, inverted repeat palindromes are more common and have greater biological importance than mirror palindrome. Main thing is that whatever the enzymes, most of the enzymes that are known, they are recognizing reverse palindrome sequencing or inverted repeat palindrome sequencing rather than the mirror palindrome sequencing. This is the simple this is the property of your recognition site. So now comes the type of restriction enzyme. So we mentioned that this is the restriction site where your restriction enzyme is going to bind and going to produce cut. But where they are going to produce a cut. So like if I'm going in the, so just I'm writing a sequence what I will written in previous slide. Okay, so sequence we have written G A T T C. So here G T T A A G. So both are the reverse palindrome GAATTC, GAATTC. So now the simple question is there. So we found that this is the recognition site. Now on the basis of where it is producing cut, near it or very away from it. On this basis, we characterize your restriction enzymes in 4 major class. Okay, so this is not just one criteria, other than these are also some criteria are also there. What are those criteria? One criteria is on their composition. What are their composition? Composition means how many subunits are there? 1 subunit, 2 subunit, 3 subunit, 4 subunit, 5 subunit, or single subunit, one property. Second, cofactor enzyme requirement, what it needs to produce a cut. Then third, nature of their target sequence, what type of the target sequence is there. and the fourth is the position of their DNA cleavage site relative to the target sequence. Means this is the target sequence where it has to produce a cut, but is it producing cut here itself, or it is producing cut 20 base pair away from it, or 100 base pair away from it, or 5000 base pair away from it? Now you may ask things are, when they are not producing cut here, they are producing cut a 5000 base pair away from it, then why are you calling them as a restriction enzyme? Can you say that? Can you answer it? These enzymes are not producing a cut which is the recognition site, they are producing away from it. Still then we are calling them their restriction site or their recognition site. Yes, Manaswini? Yuvraj?
Sir, I don't think I understood your question.
My question is there, I mentioned that we are characterizing the restriction endonucleases on the basis of first, their composition. It's well understood, different compositions may be there, you call them different one. Co-factor requirement may be
Right.
different, nature of the target sequence may be different, but this fourth one, position. Co-position means if this is the restriction recognition site, then your restriction enzyme not producing cut here. It is producing cut very far, 1000 base pair away from it. When it is not producing a cut at its recognition site, still we are calling it as a restriction enzyme. Why?
Sir, I'm just guessing. I don't know if it's the right answer, but
Yeah, I need
it's,
gas only. I need just gas only. I don't need a.
maybe they're just I don't know, leaving enough space. I mean, if they cut it too close, then what if by mistake some of the base pairs also cut? I'm not sure.
No, the thing is that why we are calling them restriction enzyme, because they, yes, they are producing cut very away from their recognition site, but if the recognition site is not going to be there, they won't produce any cut. That's why they are specific in nature. Restriction, their recognition site must be there. That's why we're calling them the restriction enzyme. The property of your normal endonucleases, they are producing cut within your DNA. They don't need any recognition site. Just add them, they are going to produce a cut. But for the restriction enzymes, there must be a recognition site. Whether they are producing near the recognition site or very away from the recognition site, that's a different thing. But recognition site is going to be there. Okay. So on the basis of these four properties, one, their composition, second, enzyme co-factor requirement, fourth, nature of their target sequence, fourth, their location position where they are going to produce cut. We are characterizing them in four different categories, and basically even four are not of we generally categorize in three. Fourth one is also a newer one, but mainly it is three: type 1, type 2, and type 3. But now we are creating type 4 also. Okay, so DNA sequence analysis of nesting enzyme, however, show greater variation indicating that those are more than 4 types. So they are not just 4, they may be more than 4 is there, but we generally we are characterizing in the 4 only. All type of enzyme perform a DNA sequence and carry out endonuclease activity due to specific fragment with 5' phosphate. So now one more thing, where they are producing cut, they are producing a phosphodiester bond. So when they are producing a phosphodiester bond at the 5', they are leaving a phosphate group. So now you may understand that if you are aware about the structure of your DNA, at the 5', the phosphate group is there, and now this 5' phosphate is joining with your 3' OH. So when they are producing cut, they are leaving a 5' phosphate and 3' OH. It is not like that they are producing a cut in a way that phosphate group now present at a 3' OH. Okay, so phosphate group is always going to be present at the 5' of your ribose. Okay, So now coming to the one by one, their characteristics, what are their specific characteristics and why we are calling them. So first coming to the type 1. So here you may also say that survive from type 1 and type 3 are going to be a similar type. We bring in between type 2. So thing is a bit different. So, but this type 1, type 2 and type 3 these are numbered on the basis of how they've been discovered. So there is no another classification in them or no other specific thing to mention that why you are calling them type 1, type 2 and type 3. So type 1 is the one which is discovered first. Okay. So what is the type 1? Type 1 is the first to be identified and were first identified in two different strains of E. coli, that is the K12 and B. So E. coli B and K12 strain, these restriction endonuclease that we are calling the type 1. And every enzyme has enzymatic nomenclature. So for that name is the EC3.1.21.3. This is enzymatic nomenclature. You can read your enzymology to find how you are naming or numbering them. So these are the first three is the class, which class it is. So nuclease are being put in the class number 3. That's why 3.1 is there. Okay. So as I mentioned, you are recognizing them on the basis of first their cleavage site. So what type of cleavage site is? Cleavage site is remote from the recognition site. So this is the fourth point. The fourth point what you mentioned. So the position of their cleavage site. So cleavage site means where they are producing cut. So recognition site is a site which it is recognizing, but where it is producing cut, that may be different. Whether it is producing within it or outside it. Okay. And when we are saying recognition site is the 4 to 8 nucleotide, it doesn't mean it is removing that 4 to 8 nucleotide fragment from the your DNA. It is producing cuts there. It is just breaking one phosphodiester bond, one in one strand, second in the second strand. So each strand is going to get one break. Not there are not going to be the two break in your DNA sequence. Okay. So here we are. So the cleavage side is remote. So now the question is how much remote, how much far is there? So enzyme is going to cut at a site that differ at a random distance. So distance is random. So here the randomness is there. Specificity is there in terms of recognition. If recognition is not there, it is not going to produce a cut. But the cut may be a bit random. So how it is producing cut? It is producing cut at least 1000 base pair away. Okay. So like if you are, may say that your recognition site is in the Noida and you are producing cut in the Gurgaon, that is the type of thing. So 1000 base pair away, it is producing cut. Then comes the second thing, cleavage at these
Excuse
random
me, sir.
site for the yes.
Sir, what is the remote site?
Remote means away.
Oh, okay.
Where you are locating, something is locating away from you, that is remote to you. You are using a remote TV remote.
Yes, sir.
TV remote. You are not pressing the button of TV with the remote. You can handle it from a distance. So here this remote in the same meaning is here. If something is present at a distance, that you call the remote. Even in case of those are in the northeast and Jammu Kashmir, we call that this a remote location. Even some location in the village, you call it the remote location, where you don't have much of the transport. So in a location which is far from you, we call it as a remote location. So here also the same thing is there. It is producing cut at a remote location. Remote location is a location which is away, 1000 base pair away from the your recognition site. Okay. So
Yes, sir.
cleavage at these random sites follow a process that called the DNA. Yes.
Nothing, sir.
Hmm?
I just said that I got.
Okay. Okay, so what is there? There is a DNA translocation is there, which shows that enzymes are also molecular motors. So translocation means your DNA is your enzyme is moving from one location to another location. So this is a, it is a molecular motor also we are saying it. Then the recognition site is asymmetrical and is composed, recognition site is asymmetrical. So what this asymmetry means? Means at one strand it may be of 3 nucleotide long, at another strand it may be of 4 to 5 nucleotide long. So both the strand, both the strand recognition sequence are not similar. So that's what asymmetrical. In previous example, we were talking about your recognition site. We are saying GATTC. So GTATAC. So both are same sequence. So if the 6 sequence are in the one strand, 6 sequence are in the another strand. But here what is there? Here there is a asymmetry. So might be in the recognition site in that one strand it is going to be 3 to 4 nucleotide, another it will be 5 nucleotide separated by a non-specific spaceship of about 6 to 8 nucleotides. So this is an asymmetry. Now clean only one strand. This is another one. What we mentioned? If you say the 4 property we said, 1 cleavage site. So 2 strand they are cleaving, 1 strand they are cleaving. So it is cleaving only one strand. It is not cleaving both the strand. Okay, so this is the property of your type 1. Then coming to the second thing, requirement of cofactor. Yes, it needs the cofactor. What type of cofactor it needs? It needs magnesium ion for its activity, it needs ATP for its activity, and also SAM, S-adenosyl L-methionine for its activity. So if you didn't give these 3 enzymes, 3 things, divalentine, magnesium, ATP, and SAM, then your type 1 enzymes are not going to function. So this is the one when we are talking about in our lab that if you add PTA, then enzyme will become non-functional. So this is because these types of enzymes, these nucleases, they require magnesium ion as a cofactor. This is the one cofactor. Other than the requirement of this type 1 is 3. They need magnesium ion, they need ATP, they need SA and SAM. Okay. Then what was the next property? What is their composition? Type 1 are having the multi-subunit. They are going to 4 subunit. HSDR, HSDM, HSDS. So these name of these that we are talking about is S, R, D, or M. They are depending on what type of function they are going to do. This means they are going to be multifunctional. So one enzyme that you're talking about type 1, it is going to do more than one function. So what are more than one function? There are the 2 types of function when we're referring to the restriction molecules. One is the restriction and second is the modification. So type 1 enzymes are having doing both the type of function. One, they are doing both methylation and the restriction. Okay, so 4 subunits that you are having, so what type of the 4 subunits they are going to do? They are going to have one HSD is the name of the enzyme. It is going to have R stands for recognition. So this subunit play role in the recognition of your recognition site. Then coming to HSDM, M stands for modification. So it is going to play a role in the methylation. Then HSDS, S stands for the your SCM binding site. Okay, so this is the thing that it is going to be playing a role. How it is going to work. Okay, these enzymes, as I mentioned, that they are producing very far, so they are not of much use in your recombinant DNA technology because when you are analyzing something, you want to produce a specific location. But they are the enzyme, the restriction enzyme which were discovered first. That's why we are talking about these and putting emphasis on these enzymes also. Then as I'm skipping the type 2 and coming directly to type 1 because when we are moving, we are just moving according to the property. So now the type 2, 3 enzymes are there. So now you may list as a type 1, type 3 and type 2. So type 3 is coming in between type 1 and type 2. What it means? The distance that you are talking about, that we mentioned, it is cutting at a remote location. So now remoteness is being decreased. So earlier I mentioned that your enzyme is recognizing Noida and producing cut in the Gurgaon. Now you decrease the distance. Now you may say that enzyme is sitting in the MAT and producing cut at the metro station, Auckland Century metro station. So now distance is a bit less, but it is still at a distant. But distance is not as large as in case of Delhi, Noida to Gurgaon. So the cleavage site, first characteristic on which these enzymes are differing from the type 1, cleavage site is short distance from the recognition site. Recognize two separate non-palindromic sequence are inversely oriented. They cut DNA about 20 to 30 base pair away. For type 1, 1000 base pair away. Type 3, 20 to 30 base pair away. So this is the one. Recognize short 5 to 6 base pair long asymmetric. So here also asymmetry is there. And cleave 20 to 25 base pair downstream to leave short single stranded 5' protrusions. So they are missing cut at the 5'. They require presence of two inversely oriented unmethylated recognition sites. So your recognition site must be unmethylated. If your site is going to be methylated, they are not going to produce cut. Okay, for the restriction dice. So these are the property of your recognition site in case of your type 3. Then the requirement, as we mentioned that in previous case, they require the divalent ion. Here the requirement of divalent ions are not there. So what they need ATP, but they do not hydrolyze it. They need SAM, but they can function without SAM also. Okay. So those are things that were must for the activity of your type 3. They required ATP for their activity. They are hydrolyzing ATP also. They required ATP, but they are not hydrolyzing it, and they don't need SAM. But if you give the SAM, their activity is going to be enhanced, but they can function even in the absence of SAM. So this is the property of type 3. Then coming to their complexity, they exist as a part of complex. So the complex is there with the modification methylase. So it means the same one modification and the recognition property are going to be there. They are a component of prokaryotic restriction modification mechanism that protect organisms from the invading. They are also a part of the defense mechanism. Okay. So now next is the most important one, that is your type 2, and these are your engineers. Whatever the type of enzyme you are going to study in detail or you are going to talk about in your laboratory, they are going to be class of type 2. Okay, so they what are their property? Their property, you are first start from the away, then come bit near. Now you reach within the your cleavage site, your recognition site. So what their type of enzyme they do? They cleave within or a short specific distance. So short means just one nucleotide or two nucleotides away from it. Okay, so most of them are cutting within or near the recognition site. Okay. The cleavage site is palindromic in nature. Palindromic means it repeats, means both the sides same. Reverse palindrome or mirror palindrome monotype may be there. So cleavage site is 4 to 8 nucleotide in length. They recognize and cleavage DNA at the same site. So your cleavage site and the cleavage site is going to be same. These enzymes cleave the phosphodiesterase bond of double helical DNA. So they are going to cut in double helix only, not a single stranded one, and they are going to produce cut at both the ends. Okay. So now at both the end, how they are going to produce cut, that will depend upon where they are going to produce cut. If they are producing cut at the same site at both the end, then they produce a blunt end cut. But sometimes they are producing cut at a different one. Then they are going to produce a staggered position or leaving a overhang. So what type of the staggered position is there, and what type of your things are there, we'll discuss detail in our next class. So now I think so requirement is there. So they don't need ATP, they don't need SOM, but what they need, they require only Mg2 divalent ion. They required, that is a cofactor. They have the only single function. They don't have the second function. They have only the function of the restriction digestion. They have the independent enzyme for the methylation. So in the type 2 system, you are having a separate restriction enzyme and separate modification enzyme. So not a single enzyme is not going to produce a cut. So this is the property of your, these are the property of your restriction type 2 enzyme. Okay, so with this I'm stopping it today, and if you have any specific question from today's lectures, please let me know.
Sir, how methyl transferase protects the endogenous DNA in the prokaryotes?
See, methyl transfer a methyl group. Like cytosine is mainly methylated. So if the cytosine got methylated, then in earlier time this methyl group is not present, but now it is methylated. So what happened now? Like it is like this. Now this is, you can say this is a nucleotide, and now this is methyl group. This is enzyme. Okay. So if this methyl group is not present, then this is there and at this location. But if this methyl group is there, then what is going to occur? Now this methyl group will not allow this enzyme to reach to its recognition site. So with that, this enzyme won't reach it here, and now this its site, its substrate changes. So if substrate changes its shape, it won't act on it. Okay, so this methyl modification changed the substrate, so that substrate is now not fit to the active site of your enzyme. That's why it got protected.
And sir, you just said the type 2 enzymes are independent of methylase. What does that mean?
Independent of?
Methylase. It's written.
Yes. So thing is that if the methylation at what location is there. So if I'm going to the previous slide. Okay, so their recognition site are different. Their recognition site is different. What is your question?
Sir, I said in type 2 enzymes, you said they are independent of methylase. What does it mean? Type 2. Type 2.
This is the okay. Type 2 independent of methylase means they do not have the methylase activity. Different enzyme is going to be there.
Okay, sir. Thank you, sir.
Means the restriction enzyme is not going to have methylase activity in them. They have only one function, that is a restriction, not the methylation. So for the methylation, you need another enzyme that is going to methyl transferase. An independent methyl transferase is going to do that job.
Okay, sir. Thank you, sir.
Got the point?
Yes.
Any other question?
Sir, can you please move to the slide of recognition site? I didn't get that slide. Can you please move to the slide of recognition site? I didn't get the third point of it.
Which you are asking?
The slide containing a recognition site.
Yes.
Yes, sir. The third point.
What you didn't get?
Sir, I didn't get it.
What you didn't get, that I'm asking.
I mean, sir, the entire thing what the line is about to tell.
This is a recognition site. It is the property of the recognition site. The recognition site in case of DNA simply means DNA sequence. So what DNA sequence your enzyme are going to recognize? So the sequence that your enzyme is going to recognize is generally 4 to 8 base pair long. So this is just 4 base pairs like ATT, ATTA. This is the 4. This is the recognition sequence. This is the main thing. It may be 6 also, like GTATG. it will be 7 also. You can put one more G here. So on. So what is the length? What sequence they are recognizing? What is the property of the recognition site that we are talking about? So the recognition site is simply a sequence which your enzyme is going to recognize to produce a cleavage. If your enzyme doesn't found its recognition sequence, then it won't produce any cleavage. Is it done?
Yes, sir.
Okay, so I think we can stop it now. And if you have any specific query before starting our next lecture, we will discuss that. So when I call your name, so just a moment, I'm opening your time sheet. Then okay, so keep on turning on your camera. When I call your name, Demetrius.
Present, sir.
Gurudev.
Present, sir.
Gurudev.
Present, sir.
मैं दोबारा नाम नहीं बोलूंगा. मनस्विनी.
Sir, I already said I'm present.
Present,
मनस्विनी.
sir. Present, sir. I'm present.
आदिश.
Present, sir.
आदिश.
Present, sir.
युवराज. युवराज.
Present, sir.
संजना. संजना. Absent.
Present.
नेहल.
Present,
नेहल.
Present, sir.
अतोयबा.
Present, sir.
देवांशी. देवांशी. Absent. नंदिनी. नंदिनी. Absent. क्षितिज. क्षितिज.
Present, sir.
जहानवी. जहानवी.
Present, sir.
शिवानी.
Present, sir.
शक्ति.
Present,
शक्ति.
sir. Present, sir.
रूपल. रूपल.
Present, sir.
साहिबा. साहिबा.
Present,
इशिता. इशिता.
Present, sir.
नम्रता. नम्रता.
Present, sir.
तन्वी.
Present, sir.
युक्ता. युक्ता.
Present, sir.
आयुषी.
Present, sir.
विवेक.
Present, sir. Also, so Devanshi messaged me. She's having network issues, but she's present.
Okay, so absentees are Adish is absent, Sanjana is absent, Divanshi,
Sir, I said
Nandini is
present.
absent.
Sir, Adhish said present, and
Oh.
I was saying, Adhish said present,
अच्छा
and
बोल रहा था फिर बोलते हैं.
I was saying that Devanshi is having network issues, but she's present.
Sir, Sanjana.
इशिता. संजना बोलते कि जब नाम लिया जाता है. इशिता. इशिता.
Present, sir.
इशिता.
Yes,
तो अभी भी 5 मिनट हो गए. अगली बार absent मार्क कर दूंगा पक्का.
Sir, I guess network issues.
So Nandini is absent. Rest all are present. Okay. So thank you all. Stay safe. Stay healthy.
Thank you, sir.
Thank you.